Hi, my name is Allie, Igwe, and I'm a microbial ecologist. Ecologists study how organisms relate to one another and their environment. In my case, I focus on how plants and microbes relate to each other and the soil. A day in my job is different depending on what part of the research process I'm in. Sometimes I'm out in nature picking plants and digging up soil. Other times I'm in the lab working with chemicals and pipettes. A lot of my time, however, is spent on the computer performing data analysis and writing about my work. I work with plants and soil, as I recently said. Plants provide many services to the environment and humans, such as shelter, medicine, and food. We see leaves in our salads and on our sandwiches, but what you don't often see is the plant root. Roots absorb water and nutrients, store energy for later, and anchor plants to the ground. A healthy root system is required for healthy shoots, stems, and leaves, and part of a healthy root system includes a healthy microbial community. A microbial community is just a collection of bacteria, yeast, and viruses and other organisms we can only see with the microscope. Maybe you have been hearing a lot about the gut microbiome in humans and how important probiotics are for our health. Or maybe you have been hearing a lot about viruses and diseases. Well, plants deal with similar issues. Plants have to find a way to stay healthy just like us, and the microbial community associated with plant roots is one way to do that. Scientists have various methods that can be used to study plant roots and microbes. One simple way is to collect the soil attached to the root and go from there.
Hi everyone. Alright, so we're getting started today. I'm collecting some roots for my experiment that I have running, and I'm just going to show y'all how I do that. Real simple process, kind of tedious, but it is what it is. So this is a deep pot, and this is one of the samples that I have growing. So I just like to take the tag out, and you can't see, so I'm kind of rolling the deep pot, tapping, trying to get as much of the soil out. So this soil is kind of wet, so it's kind of sticking a little bit more than usual than I would like it, but here we are. So here are the little roots, and this is how, I collect the root samples, and I will just go on to measure. So
once you have the soil that surrounds the root, also known as rhizosphere soil, you can isolate microbes, extract DNA, or use it for other purposes. We can characterize the microbial community, study its functions such as stress tolerance or disease prevention, and use that information to create our own microbial communities that can be used for growing more food or conserving endangered species.